A team from the National Anti-Vivisection Society's Special Investigations Department secretly films the delivery of beagle dogs to a laboratory. Inside is another member of the department working undercover as a laboratory technician. This film recounts an undercover operation by the NAVS into research and animal welfare inside 2 British laboratories. It is about your right to know and participate in the debate on animal research. A secret world out of control. That is how evidence has repeatedly shown the world of animal experiments to be. Previous investigations by the National Anti-Vivisection Society have highlighted poor laboratory procedures, poor controls, confusion about what the legislation requires, and inadequate protection of laboratory animals. Laboratory animals are theoretically protected by the Animals Scientific Procedures Act, but there is little protection on the face of this legislation. Instead, the day-to-day care is covered by the Code of Practice for the Housing and Care of Animals Used in Scientific Procedures, which details the way in which animals should be housed, and a group of faceless Home Office inspectors deems which experiments should or should not be allowed. Every experiment in the UK that uses animals is licensed by the Home Office. The details of these projects, even if they're paid for by your taxes, remain secret. It is often stated that only necessary experiments are permitted, and they're only allowed if no alternative is available. Yet we have shown experiments which are scientifically invalid. Experiments where the outcome is already known. Use of animals where a non-animal method is available, and that the Code of Practice is not worth the paper it's written on. An NAVS investigator first obtained a job as an animal technician at St Mary's Hospital in London, where rats, mice, monkeys, rabbits, guinea pigs, woodchucks and fish are experimented on. For rodents, the laboratory was akin to a factory farm, constantly breeding. A breeding mouse would give birth to 6 litters in as many months, and a rat almost as many. After 6 months of constant pregnancy and suckling, she would be killed. This led to disturbed behavior like cannibalism, with mothers eating their young. Animals that didn't breed well were killed. Most surplus animals were gassed with carbon dioxide. Some had their necks broken. Babies had their heads snipped off with a pair of scissors. The Code of Practice says that this should not be done to animals over 4 days old, but as litters at St Mary's were cut down once a week on different days, they could be older. Staff believed that they needed to constantly breed animals to maintain the stability of the colony. As a result, they killed thousands, killing more through overstocking than were used in experiments. In just 34 weeks, our investigator estimates that he saw almost 2,500 animals being killed or taken away to be killed because they were surplus to requirements. Life is cheap in the animal lab. Rats were given epilepsy by tetanus injections into the brain. Within a week, the animal begins seizures, which continue for 8 weeks, leaving them with behavioral and learning difficulties. Electrical readings are taken from screws permanently fixed in the animal's brain. There are fundamental reasons why this research should not have been permitted. The rat is a poor model for human epilepsy. The type of drugs being studied are already known to give different reactions in rats and primates. In March 1992, the Wellcome Trust awarded the team responsible a grant of £37,777 for studies of brain disease in mice. Rats are also used in kidney research. Unfortunately, not only is the lab animal version of the disease caused differently, but it causes different symptoms. Why was this research permitted when it would be far better undertaken using human material from renal biopsies? These were introduced over 20 years ago and are routinely carried out in cases of suspected renal failure. Kidneys replaced during transplantation could also be used. St Mary's Renal Unit told us that they perform about 50 transplants a year, so a wealth of human material is available. During 1992, these researchers were awarded £64,866 by the Medical Research Council, taxpayers' money. Other suffering is accidental. A regular problem is something called run-outs, where a water bottle leaks or completely empties into a mouse cage, all too often killing a litter inside. Our investigator pointed out which bottles caused the problem and asked that they be replaced. No action was taken. There were also outbreaks of disease, for example, mouse hepatitis. In room 24, mice suffered from burrowing mites. On one occasion, several mice became ill, with bloated stomachs, and 27 were found dead in their cages. Despite this, the walls of rooms were not regularly scrubbed. Water bottles were not changed every week as they should have been, but just topped up on a Friday afternoon when people were rushing to get finished. A guinea pig lies dead, an incidental casualty of heart research at St Mary's. A clamp had been fitted to a heart valve in order to make the heart overdevelop as a model for chronic heart failure, but this one didn't make it to the end of the experiment. But heart disease in people and these animals differs. The drugs being tested are known to have different effects in different species. Human tissue is used by other researchers instead of live animals. At least one drug studied at St Mary's has been used by people for 10 years, so why is it still being studied in animals? The research is so flawed, we can only wonder why it was permitted. In the year from November 1991, this team was awarded £339,552 in grants from the Wellcome Trust and the British Heart Foundation. Several monkeys, red-bellied tamarins, were brought from a supplier in the USA to be used in hepatitis experiments. In the wild, these animals would live in extended family groups and spend much of the time grooming. But it seems no effort was made to house them to the standards in the government's code of practice, which say of primates,
all need complex stimulating environments. Housing should provide adequate space, complexity, for example varied diets and cage furniture, and opportunities for social interaction. Food items such as nuts and grains may with benefit be added to bedding to encourage foraging behaviour and reduce boredom. Most species are highly sociable and benefit from being housed with companions and should be so housed that they have opportunity for social interaction. Single housing should be avoided wherever possible. Isolation for more than a few days should be avoided.
Other recommendations are that if animals are housed singly, they should at least be able to see each other. Cages should include wooden perches, a swing, a wooden nest box, a shelf for feeding, and sufficient wood shavings to allow foraging. And the animals should have space to jump horizontally from one perch to another. The code of practice is less than rudimentary, but St Mary's still failed to action at least 7 recommendations, leaving the animals alone in a barren metal cage. Simply rearranging the cages would have enabled the animals to see each other. Tests on the tamarins included experiments with hepatitis GB virus, during which one monkey was injected with the excrement from an infected animal. Despite inflicting this painful and debilitating disease, the researchers were still unable to say whether hepatitis GB is actually a human or a monkey virus. The research is made doubly unnecessary because the virus can be grown in human tissue culture and studied there. Unlikely prisoners of this London hospital are several wild woodchucks. Woodchucks, better known as groundhogs, are ground-dwelling members of the squirrel family that love lying out in the sun and hibernate for the winter. They are only found in North America, and it was here that these animals were trapped before being sold for experimentation. The Code of Practice states that
animals caught in the wild should be kept in conditions which conform as nearly as possible to their natural habitat in such respects as light intensities, food, etc.
The housing at St. Mary's speaks for itself. Here they are used to study hepatitis delta virus, which can only survive in the body if hepatitis B is present. Unfortunately for the animal researchers, hepatitis B is largely restricted to humans, so they have to make do with something similar, but not quite right. In this instance, woodchuck hepatitis virus. If infected at birth, the woodchucks are almost guaranteed to develop liver cancers. The researchers had already studied a vaccine that had worked in rabbits, but not in woodchucks. Because this project used wild animals, one might have expected a closer scrutiny before it was permitted, yet there are straightforward reasons why this research should not have been allowed. It was reliant on different viruses to the human situation. Hepatitis delta virus can only exist alongside hepatitis B, which can be studied in human cell culture. More than 20 million doses of hepatitis vaccine have been given the world over. In effect, it is already possible to vaccinate against this virus. In the year from November 1991, the Wellcome Trust awarded this team a grant of £45,657 for a 2-year project. The team has also been funded by the Medical Research Council. From St. Mary's, our investigator moved to Toxicol Laboratories in Herefordshire, which is contracted to perform experiments for commercial companies. Several years ago, the NAVS exposed the laboratory after an undercover investigation revealed details of skin and eye irritancy studies. This time our investigator was placed in the dog unit, with a capacity for 190 dogs, and in 2 months our investigator saw the unit hold 142 animals. Beagle dogs were brought from 2 suppliers, Consort, based at nearby Ross-on-Wye, and Interfauna at Huntingdon, Cambridgeshire. These 24 puppies from Consort were destined for an experiment known as COS 44C, being undertaken for the Swedish company Confarma. They will never see daylight again. They were entombed in metal-walled metabolism cages overnight to collect urine and feces samples, where it was discovered that a blunder had been made. The dogs had been given 4 times the recommended dose of the antibiotic Noradine, and this caused internal bleeding. Before and during experiments, the dogs are kept in bare pens, with just a scattering of sawdust on the hard floor to aid cleaning rather than any comfort for the animals. No bedding material is provided, despite guidelines stating
bedding and nesting material should be provided unless it is clearly inappropriate. It should be comfortable for the species.
Previously, an investigator from the NAVS had worked undercover in the laboratories of drug company SmithKline Beecham, and we were able to obtain film of those conditions as well. Clearly, when it comes to keeping dogs in laboratories, this is the norm. Project KIR-7C involved testing the herbicide ethofumisate for the Finnish agrochemicals company Kemira. A rubber hose was pushed down each dog's throat directly into the animal's stomach, and a dose of the herbicide was injected down. But these dogs were suffering in vain. Ethofumisate has been in use for years, having received its US patent in 1972, and is sold in Britain as Nortron. Up until 1985, Nortron was approved in Britain under the non-statutory Pesticide Safety Precautions Scheme. This set out a wide range of animal tests to which new pesticides must be subjected. Ethofumisate must have been subjected to them because Nautron was granted the Ministry of Agriculture, Fisheries and Food registration number 01506. Subsequently, the voluntary pesticide safety precaution scheme was replaced, and under the umbrella legislation of the Food and Environment Protection Act, pesticides came under the Statutory Control of Pesticides Regulations 1986. Ethofumicate is also approved under this legislation. Despite this, dogs continue to suffer at Toxicol. In this instance, the Home Office granted a license to experiment on dogs when the product had already been tested on animals, the product had been deemed safe enough to pass 2 pieces of UK legislation, the product had been in use for years, so details of its actual effects on people, the environment, wildlife and so on should have been monitored, and if not, why not? Animal laboratories not only test the same products, but they cling to experimental techniques which are at best questionable. In 1991, another undercover investigator for the NAVS worked for several months as a laboratory technician at St Bartholomew's Hospital, London, where rats and mice had air injected under the skin to create an air pouch just behind the shoulder blades in arthritis experiments. The St Bartholomew's researchers themselves admitted
it is possible that the mechanisms that produce the air pouch reaction and arthritis are not the same.
Unbelievably, this bizarre approximation for arthritis has been inflicted on rodents for almost 40 years in laboratories worldwide. In 1990, 3 Toxicol researchers joined 2 other labs in a study of anti-inflammatory drugs which used the air pouch technique in rats. Various drugs were tested, including indomethacin, cyclosporin, and the steroids dexamethasone and betamethasone. In the Toxicol rats, indomethacin did not work, yet it is recognized as one of the most active anti-inflammatory drugs in people. Both dexamethasone and indomethacin have been used in patients for years. With the data from their human use, why were they retested in animals? Also at Toxicol, the anti-malaria drug Artemether is being tested for the Belgian company ProPharma. Yet its active ingredient has already been tested on people and shown to be effective and non-toxic. By March 1993, more than 1 million patients with malaria had already been treated with artemisinin and its derivatives, and it was reported that there had been remarkably few adverse effects. Clearly, the system which licenses animal research is failing to protect laboratory animals, and it can only be concluded favours the researcher. Likewise, laboratories are visited by a Home Office inspector, but are given advance warning of such visits, and so are unlikely to spot the most flagrant abuses of the system. This latest investigation has added to the body of evidence showing that animal experiments are cruel, outmoded and unnecessary, and poorly controlled. The law is woefully inadequate. Whilst laboratories remain locked and public debate stifled by lack of openness and information, lab animals will continue to suffer in research that will not benefit people. And whilst this research continues, it is people who will ultimately be the losers. It is time to unlock the labs.